And I'll get started, yeah?
Yes, thank you.
Okay, perfect, thank you. So it's a real pleasure for me to be here today, and I'm very, very happy to be speaking to you. I might have slightly over-egged the title of my talk, which is The Value of Diverse Ancestries in Understanding the Causes of Diseases. So I've quickly added a little addendum disclaimer that this is going to mostly be about type 2 diabetes. And there's a little bit of confusion for those of us who don't live in the world of type 2 diabetes all the time. So there's a distinction between the different types. So type 2 diabetes is a metabolic disease. Type 1 diabetes is an autoimmune condition. And then there are other rarer forms of type 2 diabetes that do exist. So just to clarify, we'll be talking about the metabolic condition today. So this is sort of a estimate of type 2 of diabetes prevalence globally, which is put out by the International Diabetes Foundation, called the IDF, and sort of basically highlighting global burden of disease. And what you can see quite clearly is that the region of Southeast Asia has had one of the most steep increases in diabetes prevalence over the last 5-10 years. And when you consider the sheer population size of that part of the world, a 68% increase is indeed a huge sheer increase in number of people living with diabetes. And at the present moment, the estimate is that 33% of the global burden of type 2 diabetes is from South Asian populations, both living in and outside of South Asia. So all this sort of sits in the foreground of the fact that research in South Asian populations is actually quite limited and insufficient, really. And I'll speak more specifically about genetic research. So this graph here, made by some colleagues who are now at Queen Mary, actually, is trying to highlight this. So what they've done is, in the bar plots on the right, they've sort of explained they've sort of put down the global populations of different ethnic groups. So you have Europeans, East Asians, South Asian, and so on. And the cumulative plot is sort of showing you how many individuals of different ancestry groups are contributing to genetic discovery studies, which are called GWAS, genome-wide association studies. And what you'll see is that through the last decade and a half, it's really this group in red, which are Europeans, who have really been represented in genome-wide association studies, whereas South Asians, who are 25% of the global population, represented in green in this bar plot, are barely visible in genome-wide association studies. And specifically in the case of type 2 diabetes, the latest studies were done in 2.5 million individuals in total, of whom about 1 million had type 2 diabetes. And within that, 750,000 were of European origin, and only 45,000 were of South Asian origin. So there is still a pretty big gap in genetic studies. And this is with the contribution of Genes and Health, as Moneeza just sort of talked about. So what does it matter then if there's not enough research in a population? Well, diagnosis strategies, disease management strategies are all built off of research. Increasingly, pharmaceutical companies develop medications and therapeutics based on disease pathways, and the major way in which disease pathways are understood through genetic studies. So our ability to identify ancestry or ethnic specific novel genes, novel drugs, are all very, very hamstrung by the fact that there are not enough resources in which to do this kind of research. So now we're back to the foreground with type 2 diabetes. What we know from clinical studies of type 2 diabetes is that South Asians, similar to Black populations, actually develop diabetes about 10 years earlier in their lives than white European individuals. So this is data from CPRD, which is a resource of general practices across the UK. And in this study, Alison Wright has not only shown the age distributions of diabetes onset, but is also then on the right made these plots that are really trying to characterize. I'm just getting my laser pointer going. Where is it now? Draw, no. Well, wouldn't you know, this has actually changed from the last time I used it. Hopefully you can all see my little cursor moving around. Anyway, so what Alison's done here is tried to basically compare the difference in BMI between individuals who are diagnosed with diabetes at different age groups and age-matched control populations who haven't developed diabetes but are of the same ethnic group. So what you see here is that white British populations who are developing diabetes under the age of 40 have 9 kilograms per meter square higher body mass index than white British individuals who are not developing type 2 diabetes. That same difference in body mass index is much, much smaller in South Asian populations. So, and similarly in Black populations. So the change in BMI between those who develop diabetes and those who don't develop diabetes is really not as big in South Asians. And why does that and does that sort of bear out in non-migrant populations as well, is the question. So that's what we did. We sort of looked at data from India and compared it to data from Scotland. The data from Scotland is from the sort of regional NHS data, so it's pretty thorough, and those are represented in gray bars. Data from India from two sources, the National Diabetes Survey in blue, and data from a diabetes specialty clinic, which is in red. Now, everybody in this plot is diagnosed with diabetes at a young age, so under the age of 40. So that's the first group that we were looking at from Alison's plot. And even though we've applied and what we've done then is basically applied BMI-based thresholds for normal BMI, overweight, and obese BMI. And these thresholds, as you can see here, are different in Indians as they are as is meant to be in European populations. And even with those more stringent thresholds for what you would call normal BMI in Indians, what we see is that Indians are 2 to 4 times more likely to have young onset diabetes with a lean or normal BMI, which is in itself quite abnormal and unexpected. Whereas you do see what you expect in Europeans or Scottish individuals who are of European ancestry, basically, where you have a much higher proportion of young onset diabetes with obese BMI, which recapitulates the plots from earlier. So I guess what this is all getting at is that diabetes is, type 2 diabetes is not just one big monolithic condition. It's a complex metabolic condition. And more people have been talking about precision medicine. And I'm sure many in the audience have heard this term, precision medicine. And what it really stems from is the understanding that both the causes and the consequences of a condition might not be the same for everybody. So in this case, for type 2 diabetes, what's been sort of elucidated here is that you have perhaps a whole bunch of, there we go, from a laser pointer. All right, we have a whole bunch of pathways. So you're going, beta cell dysfunction, insulin action. Anyway, that doesn't particularly matter, but those are what can be thought of as pathophysiological, etiological pathways through which you can develop type 2 diabetes. Any one of them would possibly be sufficient, but in most individuals, it's a combination of different causes that does lead to type 2 diabetes. So what you have then is these sort of 4 individuals. So the last of them, individual D, has an HbA1c, which is the sort of measure of how much blood glucose you have, is at 5%, which is a normal or healthy HbA1c. And as you can see then, from their different pathways. This is called the palette model for obvious reasons, but you can see that their genetic and clinical risk is actually quite low from all of these pathways. But if we look up at individual A, who has an HbA1c at 8, and individual B and C all are at 8, which is above the diabetes diagnostic threshold. So these individuals on paper all have type 2 diabetes, but if you look at their pathways, you'll start to see that individual A has a pretty high driving force from something called insulin action, which is happening in the liver. And individual B has a high risk of several things, but like predominantly something called insulin secretion or action, and so on and so forth. So these individuals all technically have diabetes, but because of the different causes of their diabetes, their likelihood is that the strategy for treatment, management, and prognosis should be quite different for each of these individuals. So really, the promise of precision medicine is in the delivery of the right therapy to the right person at the right time. The question is if you are only studying Europeans from a genomic perspective and increasingly from what's called a multi-omic perspective, then how do you intend to deliver, any of this in non-European populations who might not share the same pathways even? So So we'll pivot a little bit to a very simplistic explanation of machine learning insofar as it has been applied to understanding the complexity and the heterogeneity of diabetes. And really the goal with all of this is to see patterns in data. And I think what we're seeing is at least in the world of diabetes, high dimensional data is a lot of information about each individual, where you have their age, their sex, their cholesterol measures, their blood glucose measures, their liver health, their kidney health, et cetera, and so on. And you also have that information for a lot of individuals. So when you want to sort of take all that and aggregate it and start to see this palette model, machine learning methods have been applied. And they're very, very straightforward methods. Unsupervised k-means clustering applied to a bunch of very sort of standard clinical measurements, has produced a whole bunch of subgroups of type 2 diabetes. And this has become extremely exciting because for the first time we can see in individuals this heterogeneity, which was hypothesized in sort of earlier publications, certainly the image I showed you just before this one. So this like with all machine learning processes has thrown up a whole different bunch of answers. But I want to focus on one particular subgroup that was sort of consistently found across all these experiments done in different populations and different ancestries. And that is this something called severe insulin deficient type 2 diabetes. So it goes some of the way in explaining this preponderance of young lean onset diabetes, because it is characterized by onset with very low body mass index, low insulin secretion capacity, and a very early age of diagnosis or onset. And what you'll also see is that Europeans, in whom these experiments were first done, have, say, like 19% of people with type 2 diabetes of European origin have severe insulin deficient diabetes. But if you sort of scan upwards, you'll see that as you go to East Asia, there's a higher prevalence, and then South Asians have a very, very high prevalence of insulin deficient diabetes from using their clinical data. And so to some extent, this sort of explains the futility of trying to estimate diabetes risk using very standard clinical measures that don't quite capture insulin secretion, which is what's happening here. And this I'm trying to sort of recapitulate using a very standard risk assessment, a set of risk assessment variables. So what I'm doing here is trying to predict when a person will develop diabetes on the basis of their ethnicity using a very standard list of risk factors. So what you'll see right here is that I can predict 25% of the variance in when a European is going to develop type 2 diabetes, but less than 10% of when a Indian is going to develop type 2 diabetes. And that is largely down to the fact that body mass index is underperforming significantly in terms of predicting variation in Indians compared to in white Europeans. And that's really the plots you're seeing on the left. So I'll just sort of deconstruct them a little bit. What I'm plotting here is age on the x-axis and BMI on the y. And the panel on the left is Indians and the panel on the right is Europeans. In keeping with gender stereotypes, red is females, blue males, dotted lines are non-diabetic controls. So anyway, I just really want to draw your attention to three things here. One is that South Asians, or Indians in this case more specifically, who have diabetes sit at the highest BMI that they will be sitting at is sort of at the absolute lowest end of what a non-diabetic control is likely to have, who's of European ancestry. So there is a very big difference in BMI across ancestries. The second thing is that the difference between, and this is again recapitulating what Alison Wright showed in her paper in CPRD, was that the difference between cases and controls is actually quite small in Indians, where it's much, much bigger in the white Scottish individuals we were studying here. But perhaps the most important thing is this linear slope. And it's this slope really that helps you run a simple linear regression model like you have on the table on the right, and actually correctly estimate risk. Whereas what you're seeing in Indians is really not much of a slope or anything. And therefore, when you use BMI to predict when an individual is at risk of developing diabetes, you're really going to stumble when it comes to South Asians. So we had this problem. We were very aware we were seeing it. And so what we were trying to do is, well, figure out if there's better metrics we could be using. So we did this rural survey of diabetes prevalence in South India. And we screened 6,500 individuals, and we did quite a bit of screen detected type 2 diabetes and all of that. But what we did was we put everybody on a pretty basic looking weighing scale. And I guess many of us now have these in our homes. These are the kind of weighing scales where you can also get a measure of your bioimpedance. So you can get a measure of your body fat and so on. And so what we did was we used these very, very simple weighing scales, which also gave you a measure of body fat, very cheap to do. And you basically got an estimate of total body fat and percent visceral fat. And what we realized is that when we put these variables into a risk prediction model, it not only were they kind of outperforming, particularly visceral fat, outperforming BMI, but your body mass index makes much more sense in the context of how much fat you carry. And this is sort of a population level sort of recapitulation of something that was sort of first noticed at an anecdotal level in 1999. So this is something called the YY paradox. Two scientists and friends, Chitranjan Yajnik of Indian origin and John Yudkin of European origin, working together in diabetes, had the exact same BMI, as luck would have it. And I guess, knew that there was obviously something much more different about them than their BMI would let on. And so submitted themselves for these things called DEXA scans, which do like a body composition analysis. And you can see the results of the DEXA scans on the side there. What the DEXA scan showed was that Chitranjan Yajnik's body fat was at 21.2%, whereas John Yudkin's was at 9.1%. And so this sort of would help to understand why your BMI alone, which would have been the exact same for these two individuals, would completely sort of obfuscate how much fat was being carried by the average South Asian versus the average European. But we, at this point, don't have scale data on fat composition across these two ancestry groups. So can we turn to genetics then to understand why this is happening? Is this something environmental entirely, or is there a genetic basis? And how is that all driving type 2 diabetes? So I know I come at this from a very genetic sort of standpoint, so I don't want to overburden people with terminology, but there is some, unfortunately. So I'll be talking about polygenic risk scores or polygenic scores, which are basically scores to characterize genetic changes across the genome, and they can basically be used to estimate risk of diseases such as type 2 diabetes. And if you have measured polygenic scores for a condition in a huge number of people, then you can also assess if that individual sits at the lower end of risk or a higher end of risk and so on. And polygenic scores can be generated for any number of conditions or traits, and they are usually generated after genome-wide association studies, which are the studies that then will first tell you what variants in the gene in your genome is associated with a particular disease or a particular trait. We'll also be talking about something called partition polygenic scores, which I will try to explain over the course of the next slide. So as I said, these genetic studies of different conditions are getting bigger and bigger. The latest multi-ancestry GWAS of type 2 diabetes was 2.5 million individuals. And what it produces is something called a Manhattan plot, which is the plot on the left. What you're seeing here are different locations along your genome. These little dots all along are sort of clustered together here, hence not visible as individual dots, but these dots all represent different variants in your genome. And the height that they are at along the y-axis suggests how small the p-value is. So the higher up the point is, the more significantly associated that variant is with type 2 diabetes. Anyway, so these experiments have been going on for over 15 years, with increasing sample sizes and more multi-ancestry in nature. Bearing in mind only 46,000 South Asians contributed to the study in terms of having type 2 diabetes, but a much greater number of East Asians did. So what is done then is that these variants are put through yet another machine learning pipeline, which this time uses a soft clustering based approach. And they are then separated along those precision medicine pathways that we talked about earlier for type 2 diabetes. So it's now separating type 2 diabetes genetic risk variants along quite specific pathways through which you could cumulatively develop type 2 diabetes. So these names that they've been given are all actually quite sort of not informative, particularly in the case of things like beta cell 1, beta cell 2, because they're sort of just separating them numerically, but they are fundamentally different in the features that are picked up. So the obesity one is the easiest to understand. These are all genes associated with type 2 diabetes through a pathway of higher body mass index, which is sort of the main driving feature of this cluster. So basically, you're getting now with partition scores a etiological specific polygenic risk for different pathways through which you can develop diabetes. And hopefully this all makes sense. And I'll try and just sort of specifically speak about two pathways, which are going to become quite important. So insulin secretion, I think if you remember back to that palette model, this was one that came up. In the current experiment is classified as beta cell 2. So what happens here is there's these cells in our pancreas called beta cells that sort of sense when you have eaten a meal that contains carbohydrates, specifically glucose. And beta cells basically are the main cells in your body, the only cells that produce insulin. So they produce insulin, secrete it, and it's insulin that allows you and the rest of the cells in your body to absorb and utilize sugar. Without insulin, your blood sugar levels would just keep rising with nowhere for the sugar to go. And you would therefore develop type 2 diabetes. And now what we found is when we use a genetic risk score or partition polygenic score, we found that compared to people of European ancestry, South Asians have higher genetic risk of having insufficient beta cells insulin secretion, and that every standardized genetic risk unit was associated with an earlier onset of type 2 diabetes. So every unit was associated with 15-month earlier onset. So you're talking about over like a year earlier onset based on your genetic risk. And the other main pathway that we identified was around how we store fat, and it's in this parlance called lipodystrophy 1. And it's really characterized by having lesser fat around your buttocks and thighs and having more fat around your sort of central organs and abdominal region. And again, your South Asians are much more likely to have this genetic risk than Europeans. And that every standardized unit increase in our genetic risk was associated with a 6-month earlier onset of diabetes compared to the average. And so this was sort of, basically what we found. And in an older version of this experiment, we had shown this for the very first time that the genetic risk of beta cell failure was very different in Indians and South Asians than it was in Europeans. And you're seeing that in these sort of these histograms on the right. We also used UK Biobank data to sort of confirm that what we were seeing was real. But more recently, and a more thorough job was done by Sam Hodgson, who is my PhD student. And what he did was he took this whole new big experiment of like 12 new partition scores that were produced from the latest type 2 diabetes studies. And he sort of checked them against some really critical traits in genes and health. So this is all done again in genes and health study. As Moneeza said, the biggest study of South Asians with genetic data available anywhere in the world. It's specifically British Bangladeshis and British Pakistanis. And so what Sam's done here is he's looked at the association of these 12 partition polygenic scores with type 2 diabetes, gestational diabetes, which is diabetes that occurs during pregnancy, and the probability of developing type 2 diabetes after gestational diabetes. And it's really important to study gestational diabetes because it is the main pathway through which young women develop type 2 diabetes, and it's much, much more likely to happen in South Asian women than in women of European ancestry. So you can see now that there are some that sort of really in their betas really show a strong relationship, some of these pathways. And what's interesting is that it's more or less the same pathways that are causing all three conditions, which is not entirely surprising. We've started to understand to some extent that diabetes during pregnancy is really a reaction to the fact that pregnancy itself is a metabolically stressful event, and it sort of precipitates a lot of the underlying genetic risk that might exist, brings it to the fore. So, yeah, we've basically identified that in addition to beta cell dysfunction, which had been shown before, fat distribution was a major driver. So was a risk of obesity. And that, of course, as I said, just to recapitulate, young women often develop type 2 diabetes after gestational diabetes, and many studies exclude gestational diabetes from their investigations. So we understand now what's causing type 2 diabetes, what's causing early onset type 2 diabetes in South Asians when we look at this sort of panoply of genetic risk factors. But let's talk about response to medication, which we know can vary within a population group. So again, in South Asians, we plotted the response to standard diabetes controlling medications. You'll see here, for those eagle-eyed, that there's no Ozempic or any of the GLP-1 receptor agonists, because there's just not enough people taking them yet. But what you have here is a change in your blood sugar levels measured by HbA1c pre-treatment to on treatment. So everybody to the left of this red line has had their HbA1cs fall or reduce after they've initiated therapy. But unfortunately, there's individuals who sit to the right of this line whose blood glucose level actually deteriorated on therapy. And this is very, very common in South Asian populations, but it does actually occur in other groups as well, where response to medication is either suboptimal or actually adverse. And what we were able to basically uncover when we looked at this was that the genetic risk of beta cell failure, or the inability to secrete insulin, was a key driving factor in all of this poor response. And so I won't go through the specific numbers, because I guess this is not really a diabetes audience, but effectively your HbA1c levels on every one of these drugs was a little bit higher after treatment than it should have been, or than it was before. And that's really not what you want. So effectively, individuals, based on how high their polygenic risk is of beta cell dysfunction, were just less likely to respond to all of these classic treatments, particularly in the sequence in which they're offered in the UK. But unfortunately, and obviously inevitably, these individuals had much, much higher likelihood of becoming insulin dependent. And for those of us who know and have family that live with type 1 diabetes, insulin is something that has to be injected, and is life altering, and is very avoidable in type 2 diabetes. But unfortunately, if you're not responding to treatment, or treatments haven't been tailored, are not precise enough for you, this will happen. And unfortunately, this is what we observed. High genetic risk of insulin secretion problems was associated with all of these outcomes. And so there's much more to summarize, but I'll just wrap this part of the talk up to say that we've used the latest genetic techniques to develop some understanding of the specific pathways that drive early onset diabetes. We've instantly picked up on the fact that South Asians are more likely than Europeans to carry a lot of these genetic factors, and that these same factors are associated with worse response to treatment. But what I want to draw your attention to are these box plots on the right, because now, unfortunately, you don't have your genetic risk in isolation. For example, an individual could have high genetic risk of insulin secretion problems or beta cell dysfunction problems, and a high genetic risk of lipodystrophy, or storing fat unfavorably. So those individuals who have high genetic risk of both pathways are in blue here, and individuals who have very low genetic risk of both pathways are in red here. And what you'll see is the high genetic risk group are diagnosed almost a decade earlier in life. So they're spending 10 years more on average living with type 2 diabetes. And in beguilingly are also presenting with lower body mass index, so 3 kilograms per meter square lower BMI, which for South Asians is quite a lot lower, considering the difference between cases and controls wasn't that high. So these are people who are almost inevitably going to not be screened as part of like the NHS annual, over 40 health MOT screens. Like they're probably developing diabetes in their 20s and 30s and being missed. And just to say that it's not only South Asians who can have this, there are Europeans and people of European ancestry who have these kinds of genetic risk, but it's just far fewer as a proportion. And I know I've been talking a lot about South Asians, but this is also the case in East Asians. And East Asians data representation has been increasing. And as a result of this, you have a really beautifully deconstructed fat distribution partition scores, which didn't exist before when it was only Eurocentric. And you can see why, because East Asians, who are plotted in blue here and denoted by EAS, which is the genetic terminology for this ancestry group, have much, much higher genetic risk than Europeans, which are in red, who are in red here, and even higher than South Asians who are in gray. So you, as genetic risk goes, you have Europeans followed by South Asians, followed by East Asians who sit at a much, much higher level of having lower fat mass in the thighs and buttocks, lower visceral to gluteofemoral fat ratios. And then they also have high risk of something called lipodystrophy 2, which is associated with higher, very specifically higher liver fat accumulation, which is sort of demonstrated in a high level of various liver enzymes. So, so yeah, there are implications for data coming out of East Asian populations as well, but certainly the inclusion of East Asians has improved the granularity of what we can understand in South Asian populations, which is absolutely great. So I'll move on to another vignette. This is around genetic selection and diabetes pathways. So, there's this gene called XBP1, Xbox protein 1, objectively a great name, but unfortunately, not necessarily a good story. So again, I guess you're now familiar with your pancreatic islet cells, amongst which you have these, in yellow, you have your beta cells. And what happens is when you have a metabolically stressful diet, meaning you're consuming things that are very high in fat and high in sugar, your beta cells are stressed because they have to keep producing insulin. And of course, insulin is very important. As I said, it's required for your glucose uptake and how your fat is stored, and everything is really dependent on insulin secretion. But if you have too much metabolic demand, then your beta cells under stress start to kind of misfire a little bit, and you have a lot of these unfolded proteins, which is just basically clutter that builds inside the cell. And eventually, if this is unaddressed, the cell will just undergo apoptosis, which is cell death, and just shut down. And so over time, if too much of this accumulates, you will lose beta cell function. And hopefully this is now starting to sound a little bit familiar to all of you, losing beta cell function, losing the ability to secrete insulin over the life course. So I won't go into the specific sort of biology of what's happening all over here, but suffice to say that there is a mechanism through which all of this mess that's forming in the beta cells can be sort of sorted out. And Xbox protein 1 plays a pretty important role in that, alongside some other transmembrane proteins. But we'll focus on Xbox protein 1. So what it does is, I'm sure you're all familiar, you have DNA and then it gets sort of transcribed into mRNA. So we're going to talk about Xbox protein 1 mRNA. So if you have enough Xbox protein 1 mRNA, some of it will get spliced. And it's this spliced version of Xbox protein 1 that's going to then solve all your beta cell problems by basically increasing the size of the cell, clearing out all these misfolded proteins, and stopping protein synthesis for a while so that the cell can recover. And all of this is very, very important to prevent beta cell apoptosis and maintaining, like in the longer term, maintaining insulin secretion. So what we basically found was that this variant that is really important in your pancreatic beta cells for producing Xbox protein 1 mRNA, and we identified that variant, which of course took a long time, and I'm just skipping all of that. Carriers, so it's an A to G polymorphism. So carriers of G are people who have the variant and who are therefore making less Xbox protein 1. So they'll run out of it faster. Their beta cells will die earlier. For every basically single variant, carriers had higher HbA1c levels than people who didn't carry the variant. And if you were homozygous, meaning you carried 2 copies of that variant, your HbA1c levels were likely to be much, much, much higher, 8.6 millimole per mole higher. And as you'll see here, the allele frequency is low in white Europeans, higher in South Asians, and very high in East Asians. So there's once again this drift you see across different population groups of some groups having higher genetic risk than others. And I won't go into the whole of it, but really suffice to say that this effect that I just showed you is really driven in people who have young non-obese onset diabetes, who are more likely to carry this Xbox variant, and who then had even higher HbA1c levels at the time of their diagnosis. And obviously, because this impacts beta cell function, your blood glucose control is at peril. And just to contextualize this, your average medication response, like from a RCT or whatever, these drug companies will do clinical trials and so on, and you see like 0.5% reduction HbA1c, 0.8% reduction HbA1c, which is what you would expect at a population level. This variant alone in young people, and HbA1c unfortunately has two different scales, but just to make it easier for you, I'll keep this in this scale. This number, 12.8, actually roughly translates to 1.2. So the effect of this variant in young onset individuals is higher than the effect of medications is on average in populations. And 25% of Asian Indians are homozygous carriers of this variant, meaning that they're in purple, GG, whereas less than 5% of Europeans are. And we've done some very simple selection screening experiments and found that this region is under selection pressure. For whatever reason, we will investigate further. And we think we're sitting on sort of a novel target for glycemic control, but only because we've been able to access populations of different ancestries. Okay, I think I'm running a little bit over time, but I'll just, am I actually, Amanita? Maybe not.
Just about, you've got like a couple of minutes.
Okay, perfect. So I'll talk finally about diabetes diagnosis and a gene called PXO1. So this is really driven by the fact that red blood cell structure and function differ a lot across global populations. And as you can see here, this has largely been because, again, the selection pressure of malaria and the prevalence of malaria in different parts of South Asia, Sub-Saharan Africa, and South America, where because malaria was endemic, there was some selection advantages to having unusual red blood cell structures. Let's put it that way, very simplistically. So how could red blood cell conditions impact type 2 diabetes diagnosis? Well, HbA1c, which we've been talking about throughout this, is universally now, particularly in the UK, used to diagnose type 2 diabetes and also to manage it, to check your response to medication, which again, I showed you those plots. It's a very robust measure of glucose control and is very convenient because you don't have to come into the clinic in a fasted state. But this is why it's potentially problematic if you have an erythrocytic condition. So in the first example here, you have an individual whose blood glucose level is normal. And HbA1c really reflects the average glucose over the 8 to 12 weeks, the previous 8 to 12 weeks. And that's because the glucose is really attaching itself to your red blood cell. And each red blood cell has an average lifespan of 8 to 12 weeks. And so when you measure HbA1c, you're getting an estimate of how much glycated hemoglobin there is in your blood. And so in this case, this individual has a higher than average blood glucose level, but normal red blood cells or erythrocytes. And you can see that they have been diagnosed with diabetes. But if you have somebody with some kind of sickle cell anemia or an erythrocytic condition, their red blood cells turn over more rapidly, and therefore glucose doesn't really attach to them, either because of their structure or because of the turnover. And you can basically get an underestimation of how much blood glucose they actually have, and you could miss a diagnosis. So in countries where malaria is endemic, other measures of diabetes are used, like fasting blood sugar, et cetera. But there are many clinically silent conditions and carrier states that are not properly reflected. So let me just skip ahead. So this is analysis done by Ben Jacobs in Genes and Health. This is again in British Pakistanis and Bangladeshis using a genome-wide association study approach, and then a trans-ancestry meta-analysis. We are just comparing the effects in South Asians and in the UK Biobank, where there's mostly Europeans. And what we did was we basically identified a variant in a gene called PXO1, which had really only been associated in a small study of South Asians in UK Biobank. but had never really come up as a HbA1c variant or genetic variant associated with HbA1c, mostly because it's very, very, very rare in white Europeans. What we realized instead was that while it was associated with very low HbA1c, it was very strongly associated with a whole bunch of erythrocytic conditions, largely suggested of very sort of relatively high cell turnover and a very low HbA1c for variant carriers. And in these violent plots here, what Ben's tried to do is show you glucose level by quartiles on the x-axis and mean HbA1c on the y-axis. And so, and then variant carriers on the bases. So basically people in the blue group are carrying the piezo variant. And so they have, you can see across different quartiles of glucose levels, the blue group has a lower measurement of HbA1c for that given quartile of blood glucose levels, meaning that you would be basically continuously underestimating how much actual blood glucose they had. And what we basically found was that there was a 6 millimole per mole difference between homozygous carriers and non-carriers. And just to bear in mind, the diagnostic threshold for diabetes is 47 millimole per mole. So 6 millimole per mole is quite a big difference. And that this variant was really not a glucose variant. It was more a red blood cell mediated pathway that was causing this effect, which basically sort of, sorry, just to go back, this basically means that if you weren't aware that this individual who is not clinically manifesting with any other sort of problems, if you weren't aware of their genotype necessarily, you would use HbA1c and basically misdiagnose them or diagnose them too late. So I'll just wrap up here and say that I've used 3 vignettes to demonstrate that there is heterogeneity in the genetic architecture of a lot of things in the world of diabetes metabolism, particularly contrasting South Asians and Europeans. And I'm doing the European contrast only because they're so well represented in studies. And the critical issue here is that with global migration, these variants are not really isolated to certain geographic locations anymore, and we're living in a mixed world now. And that the more we do these kinds of studies, the more we can feed forward the evidence required to improve health for diverse and underrepresented communities. And just to say finally that reduction is a consequence of underrepresentation. And frankly, South Asia is very diverse, huge, and there are fundamentally 3 genetically distinct groups of people who live in South Asia. And I'll be happy to chat more about this later on. And I'll just end by saying thank you very, very much to the people who do all the work. Increasingly, I do less and less actual hands-on experiments, but this is our lovely team, and I'm sure we're missing some people here. Yeah, thank you, and I'll be happy to take any questions you have. Thanks.
Brilliant. Thank you so much, Moneeza. That was definitely a tour of diabetes and everything else. I mean, for me, who doesn't know anything about it, but absolutely very informative. Now, please, everybody, put your questions. We still have got some time for questions. So in the chat, I see that there is already one which says, do you think that fat distributions and lipodystrophy could result from increased starvation evolutionarily? Is the current generation at risk because their ancestors went through starvation?
Yeah, this is a very common question that comes, in response to this, because there've been various famines, particularly in Bengal, in very recent history. I just to say, Preeti, that evolution, the effects of evolution on our genetics actually takes much longer than the time it has been since, say, the Bengal famine to today. It's more likely that the results of more recent famines have led to epigenetic changes than that they have led to genetic changes. But historically, I think we've probably adapted to the environments in which we've lived, meaning that perhaps we moved away from a hunter-gatherer lifestyle to an agriculturalist and pastoralist sort of lifestyle, which meant that we were maybe better suited to survive droughts and things like that, which would mean that we store fat around our abdomen, and then we can slowly break it down over droughts and things. And Europeans probably were doing the hunting and gathering for a lot longer. That's just a hypothesis.
So you think that it's more kind of a lifestyle change that's kind of causing this shift as
No,
well?
I think, well, I think lifestyle does make everything, all your genetic risk more apparent. I think we have more genetic risk because of evolutionary reasons, perhaps, that occurred a lot longer ago than the famines that are in our recent memory, like the Bengal famine. I think those were probably adaptations that occurred, tens of thousands of years ago, rather than just in the last 100 or 200 years.
Cool. Great. Thank you. While other people are thinking, I have a question about BMI.
Yeah.
It's kind of, I mean, it is widely used, and
Mhm.
both kind of, it's a lot of, I guess, praise for it, but it's also a lot of criticism of using BMI as a measure. What are your thoughts? And now, obviously, you have used it as a measure as well. But
Mhm.
then I thought it was actually very interesting that you also had these other measures that are kind of more informative than BMI. So, what are your thoughts on that?
In the long-term use of
Yeah.
BMI. Oh, yeah. I mean, um, yeah, I think if we can, I mean, I know the realities of the NHS are very different from like us blue sky thinking here, but I'll just allow myself this luxury. If we're weighing people on a weighing scale and getting a very, simple measurement of their weight and keeping track of that over time, and if that weighing scale can just be replaced by a weighing scale that can give you a very simple bioimpedance measure of fat mass, and we can use that longitudinally, I think it might give us a bit more information about South Asians, because that's really what we've done in that experiment is we just
Mm.
use one of those really simple but kind of bathroom style weighings, slightly more complex because it has those little hand paddles. But yeah, I can't imagine that's hard to put in a clinic, and just use, because I think the part of what, and going back to Preeti's question, part of what makes BMI so uninformative in this fat distribution story is the fact that we have more fat, but then what you're missing is the muscle mass, right? Because you have the
Yeah.
same, you have the same BMI, in the case of the YY paradox. So yeah, I mean, just I think it's more important to track that change in fat rather than to track change in weight, if that makes
Yeah.
sense.
No, no, definitely. And I think, yeah, as you say, yeah, it's the fact that the bone mass kind of just gets
Mhm.
lost in that, in that measurement as well.
Yeah, yeah.
Another questions from Phil. Very interesting information. Thank you. There is no genetic testing for type 2 diabetes in the UK. So do you think there needs to be one?
I think I think particularly if you're in a family with a high prevalence of type 2 diabetes, then I think it does make sense. Because of course if it's a genetically inherited risk, you're even more likely to have it. I know, again, the reality of having and scaling out type 2 diabetes, or any kind of genetic risk assessment is complex. But as you can see, there is a subgroup of people who would really benefit from it, because I think by the time they get scooped up into the NHS system with the diagnosis, it's probably a bit late for them, or they're probably symptomatic, which is worse, because that means that you've had high blood sugar levels for a while, you're most likely developing eye disease, and it's pretty awful. And I would say that, I guess, yes, in particular ethnic groups and in particular in families, particularly with a high risk, it should be done. There should be genetic testing. More importantly, I think the opportunity we have really is to think of what are the medications you give people who have these, what could be thought of as atypical for the completely Euro-driven norm, atypical type 2 diabetes. And it cannot be metformin as a first line of treatment, basically, which is the standard of care. And I think there
Commonly.
has to be an adaptation on that front.
Sure, thank you.
Yeah.
More questions from Preeti. Thank you, Preeti. The
Yeah.
TCF7L2 variants, the beta
Yeah.
cell 2 and
Yeah.
the type 2 diabetes discovery were from population in the States. I'm not going to pronounce the
Yeah.
name.
Yeah. It's fine. I got it. Yeah.
Did you find any more prominent gene variants in the South Asian population?
Yeah, I mean, TCF7 is like one of those absolute classic type 2 diabetes risk variants that also came up in Europeans. It's that important, even though it's a beta cell function variant. What happens, Preeti, is very often because of the genetic architecture between ethnic groups, you'll find if it's in the same gene, you might find different variants in different ethnic groups that are causal or statistically most associated. But we know the function and how TCF7 like 2 works. And so I guess the question is are we in a position now to identify and discover new genes that are potential therapeutic targets? Because TCF7 like 2 is well established and well known. So our effort now is to find new stuff like the Xbox protein stuff that I was showing you about, you know. So that would really be the goal.
Cool. And then the final question, also from Preeti. Are the South Asians more prone to type 2 diabetes and hypertension or any other metabolic problems potentially?
Um, yeah, they're more, unfortunately, more prone to heart disease as well. I'm not entirely sure if I know the answer to the hypertension question, Preeti, but I am pretty sure about the heart disease thing. And South Asian men in particular have very, very low good cholesterol.
Mm.
And so yeah, so they are even more likely to develop heart disease and stroke and so on. Yeah, sorry, that's a
Sure.
lot of bad news for us South Asians.
All right. And finally, Phil says there are many new initiatives in development looking at pharmacogenomic testing.
Mm.
Would uh, this be a better than genetic testing?
Pharmacogenetic testing is only relevant if you're getting that particular medication. So this is happening more and more with like clopidogrel, which is a drug for strokes, and which is, you know, and it's very important in South Asians who carry a variant that makes clopidogrel kind of useless, and then you're more likely to have strokes. So so pharmacogenetics is very specific of do you need this drug and then are you going to properly metabolize it? And if you are going to be taking a drug, then yes, you know, having testing before it is useful. Disease testing is more important in terms of you could, if you are screened and you identify that somebody's at risk of type 2 diabetes, you could apply interventions early in their lives to the point where perhaps they don't develop type 2 diabetes. And it would be a dream scenario to work towards that, you know, like okay, we have these polygenic scores, we understand genetic risk enough now that we can, you know, your genes don't change from birth. So if we can measure this early on in your life and give you the right interventions, whether it's dietary or exercise or whatever, to the point where you never develop diabetes, never have to live with that or manage that condition, that would really be the best, in
Mm,
my view, Mark.
great.
Mark, sorry.
Yep.
Phil. Phil, sorry.
So, yes, thank you. And Mark, a lot of questions from Mark, so I'm just going to merge the two. So Mark is wondering if there are other databases that you can recommend because I have found some interesting discrepancies in existing
Mhm.
resources.
Yeah.
Uh, and what your recommendation would be. And in addition to that, your thoughts on using minor allele frequency, of course,
Mm.
as commonly used, which is of course has got relevance in clinical trials and so on.
Uh, yeah. Um, I think I'll to your first question, Mark, there are biobanks now in East Asia, a Biobank Japan, Korean Biobank, that have front-facing information on genetic aggregation. So you could use those, and I think that's a good place to start looking. In terms of your second question, I think if a genetic variant is important in the functioning of a protein or a gene and it's absent in one entire population group, like PXO1 is monomorphic, as we call it in Europeans, then it doesn't make it redundant. It just means that defect only is occurring in proteins in East Asians or in South Asians or whatever group actually have that variant. And it makes MAF testing actually very important because if you don't carry the allele, the minor allele, the variant allele, then you're not at risk. So it's really our only I don't know if I misunderstood your question, but yeah, if it's rare, then and if it's causal, then you don't have it, you don't have to worry. If you have it, you should probably try to do something about it, or so I think, yeah.
Great. Thank you. Okay, I think I'm just going to stop for this now. Subra, I am very sorry, but if you could just send that questions to Moneeza, and that will be great, because we've just struck the 5 o'clock.
Yeah,
And
sorry I ran long.
it's been really, really amazing talk. Thank you, Moneeza, for your time and your effort, and for everybody's questions and engagement as well. Thank you, everyone. I'm going to close this webinar now. So, but as Lizzie put in the chat, next time our next speaker is Segun Fatuma, who has actually been working on genomic diversity in genomic diversity in African populations. So please look out for the next talk. Thanks, Lizzie. That will be coming in June. Thank you, everyone.
Bye, guys. Bye, Amanita. Bye, Lizzie.
Bye.
Bye.
Bye.